SegmentNT is a versatile foundation model for genome annotation. It operates at single-nucleotide resolution, processes sequences up to 50,000 bases long, and locates 14 classes of genomic and regulatory elements in a single pass, including key gene features like exons, introns, and splice sites. SegmentNT generalizes well to unlabeled genomes. It performed strongly on 17 species in testing, and a fine-tuned multi-species version further improved accuracy on distant genomes, without sacrificing performance on human. SegmentNT has the potential to open the door to flexible, high-resolution genome interpretation across species, tasks, and contexts. From isoform prediction and variant assessment to multi-species annotation, it brings scalable genomic insight closer to reality.